Hello. This mini lecture is to accompany Chapter 12, which goes over DNA technology. And so there are going to be three of these little mini lectures, and this first one is the basic tools of DNA technology. And so let's jump right in. So I want to remind you that all living organisms, all cells, eukaryotic, prokaryotic, bacteria, plant, animal, fungi, all cells use very similar mechanisms to pass their inherited factors to offspring. They all use DNA, and these inherited factors are converted to physical traits in the same way in all cells. All cells use the DNA to make a copy in RNA, and then that RNA is a message that's taken to a ribosome, and using transfer RNA, it's made into a protein. All cells use the same genetic code. The same three bases are converted into the same amino acid. Okay, that's really an important idea, and it means that if you take a gene out of any one organism and put it in another organism, it's going to be made into the same protein. And all cells also use the same enzyme and same processes to copy DNA. They separate the DNA strands into two strands, and then an enzyme, a single enzyme, DNA polymerase, adds complementary bases to each strand. So each strand acts as a template. These processes, okay, making proteins from DNA and copying DNA, can be made to work in test tubes. Actually, it's pretty easy to make it work in a test tube. And in fact, you can automate the process. You can have a machine do it. And so this is kind of the basics of DNA technology. There's a couple other important kind of things, but it's all similar. It's all stuff that was discovered in cells. One additional thing was that both enzymes that handle DNA and specially made DNA probes can find specific sequences of DNA and bind to them. And so these probes or these nucleic acid or these proteins, if you put them in with a mixture of single-stranded DNA, they can find the exact sequence and that they're supposed to work on. And so that's really neat. And so it can help us find a gene we're interested in. It helps an enzyme find what it needs to get going. One additional important type of enzyme that's needed in DNA technology are called restriction enzymes. These are enzymes that were found in cells. And restriction enzymes, they cut DNA. They find a specific sequence and they cut that DNA at the sequence. Okay, we also have another enzyme called DNA ligase that works to heel cut DNA. And so we can cut and splice DNA together, really an important thing, and I'll say more about it later. Another important discovery has been that some cells and viruses have specific DNA sequences that are useful to be inserting genes in other cells. These come from bacteria. Those are called plasmids, and this is a picture here of a plasmid. Plasmids insert themselves from one bacteria to another, but viruses can do similar things. And so if we cut a plasmid or cut a phage piece of DNA, we can often insert a gene into it and then insert it into another cell. And so we have ways of not only mixing, cutting and pasting DNA, but inserting DNA into cells. Okay, so DNA, the summary here is DNA is the same chemical in all cells. That using enzymes and other chemicals, we can find and manipulate DNA. And we can do it in test tubes, and we can do it in automated systems. These are important features because not only can we figure out now what organisms are doing, we can use these things to identify and copy DNA samples, identify DNA sequences, and move genes from one cell to another. And so the basics of DNA technology are things we discovered from cells, and now we can harness them and use them to move genes of interest and find genes. And so we're going to go over a couple of applications of this in the next mini lectures.